All right, so welcome back here today on this Friday. So what we're going to do is we're going to finish up the last few things about enzymes, and then we're going to talk about ATP or adenosine triphosphate today, because starting next week we're going to be looking at the processes of photosynthesis and cellular respiration, and ATP is really important or really key to understanding both of those. So, where we had left off last time, we had finished up with looking at different factors that affect the activity of an enzyme and their rate of an enzyme catalyzed reaction. And we're just going to finish that up today, talking about some uses of enzymes in biological systems. Um, so this picture didn't really turn out the way that I had hoped, but the idea is that a lot of enzymes are coupled together in a long chain of turning one raw substrate into a product. So if we think about in between each of these steps is kind of what we call an intermediate substance. We start off with A and then there's B, C, D, E, F, G at the end. Um, One enzyme might catalyze the reaction that turns what we start with A into an intermediate B. And then B is the substrate from enzyme 2, which turns it into C. And C is the substrate for enzyme 3, which turns it into D, down what we call a metabolic pathway of linked reactions all the way till we get to a final product, or G. Right? And this happens basically in every single body system that we have. This is how we kind of synthesize our own molecules is through these metabolic pathways, and that's kind of what it looks like, right? Um, so if we look at the picture over here, here are four enzymes that are embedded into the plasma membrane. They're integral proteins, and you can kind of see how the substrate of one, ends up going in and is catalyzed to what becomes the substrate of two. And after it is broken down in two, it becomes a substrate in three, and so on and so forth, right? So it is a very efficient way of making sure that all of the biochemicals that we need are able to be made, this kind of metabolic pathway plan, and it increases the amount of control we can have over these pathways or how we regulate it, because it is able to be shut down, that metabolic pathway, or stopped at any point in that chain. Enzyme 1, 2, 3, or 4, they can all be specifically inhibited if we need one of the intermediates along the way. So the idea is that in a lot of the metabolic pathways that are in our body, um, the efficiency comes from, like we said, the organization. The fact that our enzymes are organized into groups that are embedded in membranes and arranged sequentially, kind of like this last picture from enzyme 1 to enzyme 4. Because they're organized in this way, we're able to link endergonic reactions that require a net input of energy with exergonic reactions that produce or give off energy, right? Um, Down here is an example of this. Okay, and we're going to talk more about this picture because it goes with cellular respiration next week, right? But each of these protein complexes are enzymes that help catalyze certain reactions, all right? Another thing that we had talked about just previously is that um, organizing metabolic pathways and these sequential steps of enzymes increases our ability to regulate. So, for example, since each product of the previous step is used as a substrate in the next step, the overall product that we get at the end often acts as an inhibitor, like we talked about last time, that inhibits the activity of one of the enzymes in the previous steps. So G in this picture ends up being a non-competitive inhibitor, also called like an allosteric inhibitor, of enzyme number 1 that prevents the overproduction of G. And this is what we call feedback inhibition. And it allows greater control over this pathway because we don't just unnecessarily accumulate G when maybe we don't need to make any more. So when G is being used up by the body really quickly, it isn't able to hang around and inhibit enzyme 1. But if we're not using G as fast as we're producing it, it'll eventually build up and be able to inhibit enzyme 1 and then downregulate, we call it, the production of itself. So as an example, here is a metabolic pathway or a biochemical pathway that shows how isoleucine, the amino acid, is synthesized from threonine, another amino acid. Okay, so if we have threonine here, there are a number of steps, each with their own enzyme that catalyzes it turning it into isoleucine. However, isoleucine ends up being an inhibitor that blocks or inhibits enzyme 1 from uh, turning even more threonine into isoleucine. This way we can balance, all right, the relative concentrations of both amino acids in the body. All right, so that's just where I wanted to end with enzymes. I didn't want to put this in yesterday's video because I think that went on long enough. Um, but now we're just going to kind of take a pause and we're going to switch gears and we're going to start talking about ATP. Right, this enzyme stuff is just going to come back, but we're going to take a pause and talk about ATP today to get set up for beginning cellular respiration on Monday. Oh, forgot that was in there. So ATP, or adenosine triphosphate, and here is a picture of it, is a very important molecule. It is what cells use as energy. So when we ever talk about a process needed energy or produced energy, ATP is what we're talking about as that unit of energy. So organisms, all living organisms are endergonic systems, net endergonic systems. In total, we need energy to be put into us consistently to keep us alive, right? We need it to make new biomolecules, to reproduce, to grow, to move, for our cells to do active transport, all right, and to keep as warm-blooded endotherms, keep our body temperature stable. So we need a net input of energy. That's why we eat and break down the energy in food, is because if we didn't have a net put in energy, we wouldn't be able to survive. So I know we talked about this a few days ago, but this work that we talked about on the last slide, what we need energy for, we get by energy coupling. We use exergonic catabolic reactions like in digestion, where we're splitting atoms apart via hydrolysis and producing energy. We use that energy then in endergonic reactions, to power synthesis reactions, to build new complex molecules that our body needs. So that's why when we ingest taking in energy, eating organic molecules like carbs, lipids, proteins, nucleic acids, our body first breaks them down in digestion, a catabolic reaction, to capture the energy that's being released from those organic molecules we're breaking down. And then we need to use it in endergonic reactions, anabolic reactions. And we need what on here it says an energy currency, a way to move energy from where it's being released in catabolic reactions into where it's being used up in anabolic reactions. So we need some sort of short-term way to store it until we can immediately use it again in anabolic reactions. And ATP is really that link. It's a way to temporarily store energy as chemical energy in bonds between exergonic and endergonic reactions of the body. So ATP stands for adenosine triphosphate, right? And that's kind of a space filling model at the top, and there's more of a line drawing structure on the bottom. One of the things that you'll notice right here, this part, it's basically an adenosine base like an RNA because it has an adenine base, it has a ribose sugar, all right, and a phosphate group. That would be an adenine nucleotide in RNA. But we just add two extra phosphate groups. So its structure is very similar to a nucleic acid nucleotide. So that would be the nucleotide right there, and it just has two extra phosphate groups. So we can take this nucleotide, which we can call adenosine monophosphate. There's one phosphate. We could add another phosphate to it and make it adenosine diphosphate. That's what ADP stands for. Or if we added a third one, we'd make AT, adenosine triphosphate. That's the only difference between all of these three, which you'll see, is how many phosphate groups it has. The idea is when we add more phosphate groups onto this, we're adding energy. Having to put phosphate onto this molecule is an endergonic process. Just in the same way that if we would break phosphate groups off, it would be an exergonic process. So hopefully now we're starting to see how energy can be stored in ATP. So if we start with adenosine monophosphate right here, and we add another phosphate group to make adenosine diphosphate, and then we add a third phosphate group to make adenosine triphosphate, we are in total putting chemical energy into this molecule. Those bonds between the phosphate groups are very high energy. It's difficult to add each negative phosphate group on to the last. And so those bonds become storage houses for cellular energy. So kind of like it's saying here, each of these phosphate groups become harder than the last to attach. So the bond between the second and the third phosphate contains most energy. The most energy is stored in this bond right here because it's hardest to keep it bonded together. So you can think of this kind of like a spring. When we're adding the phosphate groups, it's like compressing the spring. And when we would take one away, it's like unloading the spring and releasing all of the energy that's stored in those bonds. So, how then do we transfer energy from ATP to other substances? So, first when we go from ATP and remove a phosphate group to make ADP, it releases that energy about, 7.3 kilocalories per mole of ATP. It's a lot of energy. That energy can go to fuel other reactions via phosphorylation. And so we took a look at this before in the signal transduction pathway and in cell cycle regulation with cyclins and cyclin-dependent kinases. So when we transfer that phosphate group to something else, we give that something else its energy, that 7.3 kilocalories per mole. All right. So that's the main way that we transfer energy using ATP is ATP phosphorylates something else, and that additional energy that's moved from ATP to the other substance via phosphorylation destabilizes that other molecule so it can become active or it can start to undergo a chemical reaction or things like that. So here's an example of phosphorylation. Building polymers from monomers in dehydration synthesis. Okay. So to run this reaction, making a disaccharide from two monosaccharides, we need 4.2 kilocalories per mole to do this dehydration synthesis reaction. So what we're actually going to do is we have an enzyme that will utilize ATP to phosphorylate both of the monosaccharides so that the energy is present for them to be able to come together to form a disaccharide. So it's more simple than just two monosaccharides and water and we spontaneously make this. We have an enzyme, a kinase, that utilizing ATP phosphorylates the monosaccharide so that they become destabilized and react, providing that activation energy to form our disaccharide, our synthesis product. And then we have the phosphate group left over that can be reused by ADP to make ATP all over again. Another example that we're going to talk about next week is the first step of cellular respiration, where we're beginning to break down glucose to make ATP. Now, kind of counterproductively, you might think, to make some ATP, we actually have to invest a little to start out with, just kind of like that idea of activation energy. And so we use 2-ATP to help these two kinase enzymes phosphorylate glucose to form this intermediate product called fructose 1,6-bisphosphate, which is now very energized and unstable. And we can use its instability to break it down into two other molecules that we can turn into ATP. So we're going to go over this in more detail next week, but I just want to insert this example here so you can have two different views of how phosphorylation is used in transferring the energy from ATP to other substances that then become destabilized so that they can go through chemical reactions. So kind of lastly, there is an ATP ADP cycle. So we can't store ATP. It's a good energy donor, but we can't really store it for very long. It's too high energy. It transfers phosphates way too easily, and we can only use it for a short time. Whereas like carbohydrates and fats are better at storing energy for the long term. The thing is, though, we can consistently use and remake ATP from ADP by using the energy from breaking down food to turn ADP into ATP, and then use ATP to power cellular work, and continue this cycle of using and rebuilding ATP molecules and linking these anabolic and catabolic reactions. So as an example, like you saw here, a working muscle cell might recycle over 10 million ATP per second. So this cycle might happen for 10 million ATP per second in a working muscle cell. So, needless to say of how important it is, ATP is very important to the cell, and so cells spend a lot of time making and reusing ATP. All right, and I think we're going to stop at that point because this then starts to get into the beginning of cellular respiration and how specifically ATP is recycled from ADP, and that has more to do with cellular respiration. So I think I'm going to hold off on that, and we're going to end here with today's talk about ATP.